In the last video, we talked about the pairing between GC and ANT nitrogenous bases, and they're very particular. We said every time we see one, we see the other, and they do not match up in any other way. This is the way that they're going to come to us. And in one route, we had 2 hydrogen bonds, and on the GC side, we had 3 hydrogen bonds. So what you're seeing on the screen right now is just basically the last slide that we ended up on in that lecture video. So I'm just going to kind of clear the screen out, zoom in on just G and C, and here you go. And there is representation of the 3 hydrogen bonds there one more time. Also, look at these little squiggly lines. That means there's more to come. There's more than meets the eye here. There's my 80s Transformer reference. As a kid, I was growing up. I watched Transformers, did you? And then on the other side, we see A and T. And again, squiggly, squiggly, meaning there are other things that are coming along here that we need to worry about, and we need to figure out what that is. So what I want to do when I start this video off is go back and talk about this pairing, and there is a proper term that we use for the pairing, and this is called Chargaff's rules. And Chargaff is basically going to come around and state something that's very obvious, of course. At least now to us it's obvious. But they're going to say there are equal numbers of adenine and thymine in DNA. So if you were telling me that every time I see an A, there's a T, and that's the only way that they pair up, then that means there's an equal number of A's and T's both in your DNA genetic code. There are also equal numbers of G and C for the same reason. If that pairing exists every single time, and no matter what happens, but those two stick together every time, then that means if I know the number of G's, then the number of C's will have to be exactly the same in my genetic code. Now, why? We'll talk a little bit later on down the road when we get to the fuller, bigger picture, but this is what Chargaff is going to say. Not only that, but trends have basically shown us that there is a difference in the ratio here. So if we look at bacteria and we only focus on the number of GC pairs, GC pairs for bacteria is 25 to 75% GC pairs. Quite a large range depending on what they're doing. But if we look at mammals, well, that window gets a little tightened. It goes from 39 to 46. You might as well round that off. 40 to 46% of GC pairs in mammals. So there could be a difference in the number of pairings that we have of GC. And this is going to lead into the discussion of what really makes our DNA different from each other. Soon we'll get to that point. But again, we're doing the build-up in order to get there first. So there is a difference on the numbers of A's and T's and the numbers of G's and C's across the board for everything. And here is one of those examples: bacteria versus mammal. Now, if you go back a couple of videos ago, you probably have taken a note, and that note had talked about a sugar. I told you that this is typically a pentose, and pentose means 5. So here we go. There's our sugar molecule. I've grayed out the other areas I don't want you to pay attention to right now, but this is our sugar molecule. A sugar molecule will be a 5-membered ring. I said 5 members, not 5 carbons. So right there's a carbon, here's a carbon, there's a carbon, there's a carbon, so there's 4. But the 5th one is the oxygen that's right back there in the back. There's your 5-membered ring. The definition of a sugar typically means that the OH group is on every single carbon on around. The problem is that this structure is modified, and we don't necessarily see an OH on every single carbon as we go around the ring. Well, there's a number of reasons for that, and that's because we have to attach these things in some form or some fashion. This molecule is a sugar. It is a 5-membered ring sugar, and in particular, it is one type of sugar, and that sugar is called ribose. So what you see over on the right-hand side are 2 versions of ribose. The first one is really the straight chain. Ribose can actually open up and go to this straight chain. Not a big deal. There's my first carbon. Here's my second carbon. There's my third carbon. There's my fourth carbon. There's my fifth carbon. That's a pentose. Notice that on this normal structure, there's an OH group on that one. There's an OH group on this one. There's an OH group on the middle. There's an OH group on that one. And then here's the ending of it, or the head of it, depending on how you look at the molecule. And this could be an OH, but right now it's a double bond O. Well, if this molecule circles around on itself, we can get this ring structure. And this ring structure is really how a lot of the sugars are presented. And this is how we draw a lot of the sugars, whether they're pentoses, whether they're hexoses, or what. This is kind of the natural form. A lot of that information doesn't go away, though. It's just drawn differently here, okay? So I still have a carbon here, and a carbon there, and a carbon here, and a carbon there. And look, the fifth carbon is kind of right there. But this is going to make a 5-membered ring. So it's still 5 members in the ring. We've just kind of changed the definition up a little bit. That's all that we did. Still 5 carbons. 1, 2, 3, 4, 5. And this ring is a 5-membered ring. 1, 2, 3, 4, and fifth is oxygen. So it's still a pentose. That has an OH, that has an OH, that has an OH, and look up here. That has an OH too. This did have an OH, this connector before it branches out. And that OH would have been right there, but that had to lose a hydrogen so a new bond could form to make that ring structure. And this will then open back up, and it will make the straight chain. And then it will revert back and make a ring. And then it reverts back and makes a straight chain. Then it reverts back and it makes a ring. And do this over and over and over. Down at the bottom, the ring structure. This is really one of the fundamental building blocks of RNA, ribonucleic acid. But think about DNA. DNA was deoxy. So what does deoxy mean? Deoxy means that we lose an oxygen. Which one do we lose? Well, here is deoxyribose. That's pretty easy to see, I hope. If I go back to RNA, it looks like it's this OH group that we are focused on. And if that OH group is there in that sugar, then this is what's going to be making up RNA. But if we remove that OH that was right here, and there's nothing there, then this is the sugar that makes up the DNA molecule, because it's called deoxyribose. And sure enough, that's what we're going to see. So if I go back to this structure that we're going to build upon, that is the term of the sugar down here below. And that is why you kind of see that not have an OH right there. There's 2 hydrogens. Therefore, this is going to be the building block for DNA. We're missing an OH group. Now, these other areas I've shaded out for you. And what are these other areas? Well, we've talked about phosphorus. Phosphorus, we said, we labeled that. This is what showed a transfer of information or a part of the virus into an E. coli cell. So we knew phosphorus was going to have to come along. Well, what you're seeing here is a phosphate group, PO4. And off of each one of these oxygens, that R just stands for a random alkyl group. That's all that it means. So we're just going to put carbons here if they want to be. They don't have to be. But we could get those oxygens attached to other things. And that phosphorus has a double bond O. So if I go back to this structure, that I've shaded out. We have a sugar. We already said a base has to be present. A nitrogenous base has to be present. And in particular, one of the pyrimidines or purines, A, T, G, or C. And now we know a sugar has to be present, but actually a deoxy form of the sugar. And now we know that phosphorus is also going to be present because we've talked about the importance of it. And all three of those pieces have to fit together. And this is how they do it. I see the ring structure that's down here at the bottom. That is my sugar compound. It's a deoxy form, which means we're talking about DNA now. Off of one end of the sugar molecule, we get this O attached to my phosphate group. Right there. There's one piece to the puzzle. We're getting bigger and bigger. And then up here at the top, I see this carbon, and there's a big long bond that goes up. They just do this so that way they can draw it away from this. It doesn't clobble everything up and make a mess for you. It's the only reason. And this is the presence of the A, T, G, or C. So this is my base that's up here at the very top. So in this structure, we have a purine, we have a sugar, and we have a phosphate group. And this is what we refer to as our nucleotide. Now, the nucleoside would actually be split off right there. The base and the sugar is my nucleoside. Then when I add the phosphate group on, that is what we refer to as the nucleotide. It's gotten bigger. We're getting ready to link these things together. And away we go with a chain sooner or later that we're going to call a DNA chain. So in this slide, what you're looking at are various nucleosides that could be out there in DNA or RNA. And I'm just going to draw a line down here in the middle all the way across. And the reason is I'm separating these into two categories. Notice all the ones up here on the top have this OH group at that number 2 spot in the ring. So this is really the presence of RNA because we have that OH group that's there. Down here at the bottom, this is our DNA. And DNA does not have that OH group at that number 2 position. So RNA versus DNA, they're almost exactly the same, right? There's no differences in the bases. There's no differences on these OH groups that are hanging off. It's just only in this one spot at what we call the 2' position. Why primes? Because this is like a second ring, and the bases are the first ring. So this could be numbered 1, 2, 3, 4, 5, 6, whatever. And then the ring could be numbered 1, 2, 3, 4, 5, 6, whatever. And because this is the second ring, we can actually denote that with a prime digit. So 1', 2', 3', 4', 5' of that second ring that's involved. So that's why they call this 2' deoxy. 2' deoxy. 2' deoxy all the way across. Okay? All right, so here are the nucleosides. Now, when we add the phosphate groups on, what we're doing is we're taking these and we're getting rid of the hydrogen. There's no more hydrogen that's there. And we are connecting that with now a phosphate group, a PO4. So we can have 1 phosphate group, we can have 2 phosphate groups, we can have 3 phosphate groups. And if you've taken a biology class, you've probably have talked about energy and this thing called ATP. Well, this is kind of where this is coming from. So this is a nucleotide, and we can actually call this adenosine monophosphate because there's only 1 phosphate group there. And this is adenosine diphosphate because there's 2 of them. And this is adenosine triphosphate, ATP, because there's 3 of them. And each time that we remove one of these phosphate groups, we can release a little bit of energy. So this, again, is the RNA version. And then down here at the bottom is the DNA version, because I do not see OH groups that are attached there. We really call them the same thing, except we say 2'-deoxy in every single case, because we have to let you know that OH group is not present. All right, so just a slight difference between the nucleosides that you're seeing here that have no phosphate at all, and then the nucleotides that you're seeing here, which does have a phosphate group. All right, so we're slowly building up. We're getting more and more complicated. So I hope you're still following along. I hope that you still are taking notes. So let's pause here. Go take a potty break. Go get you a snack. And then we'll come back and we'll pick back up from here and we'll continue to talk about the DNA structure.